Biology 0610, Paper 4, Variance 1, October, November 2020. Starting with question 1. Water is an essential molecule for life. Complete the statements. Water moves into and out of cells by osmosis. Osmosis is the diffusion of water into and out of cells through the cell membrane via partially permeable membrane. Water is known as a solvent because it can dissolve solutes. Almost all solutes can dissolve in solvent, and almost all solvents consist of some amount of water. Part C. A plant was not watered for 1 week. Figure 1.2 shows a series of photographs of the plant during the week. Okay, so this is the initial state of the plant. This is that plant after 3 days, and this is the same plant after 7 days. Explain how the lack of water has affected the support of the leaves of the plant shown in figure 1.2. Use the term turgor pressure in your answer. So, what actually has happened is that water has been lost by the plant via transpiration, and the water lost by plant was greater than the water uptake or input. Hence, this resulted in a decrease in the turgor pressure pushing against the cell wall of this plant cell. So, because of this inefficient support to the cell, it resulted in the cell wall and the plant becoming weaker, and eventually resulting in collapsing or wilting of the leaves of the plant due to inefficient or lack of water supply to the plant and lack of turgor pressure to the plant. So this is also known as plasmolized or flaccid leaves or plant cell. Um, Sorry, the camera is a bit, okay. Pathogens cause disease. Figure 2.1 shows some cells that are part of the human immune system. These cells are responding to one type of pathogen. Okay. Explain how the immune system responds to the invasion of pathogens. So basically, when a pathogen invades into your body, what happens? A pathogen is noted within the body. The body detects that yes, a pathogen has invaded the body. Then the white blood cells take action. First, the antibodies and antigens are specific in shape to each other to bind to each other, just like the active site of the enzyme, like the enzyme supplementary complex. So lymphocytes, which are the type of white blood cells that are responsible for antibody production. Lymphocytes produce antibodies that attach to the antigens, and as they attach to the antigens, they send a chemical signal, signaling phagocytes to come and produce digestive enzymes to engulf and destroy the pathogen. After the initial response, the memory cells store the instructions for this specific type of antigen for future response. This will result in immunity. And during this time, while the memory cells are working on storing these instructions, the individual may get sick for a time period, allowing for the memory cells to retain the instructions for future response, developing an immunity inside the human's body for this specific pathogen. A vaccine was introduced in 1942 for a particular disease. Figure 2.2 shows the effect of introducing the vaccine on the number of cases of the disease in one country. So this is the number of cases of disease per million people, and this is the year. So this is a time lapse from 1920 to 1980. So as we can see that before the vaccination was introduced, the cases started small and they started to gradually increase over time. Okay, but as the vaccination was introduced, there was a big drop in the number of cases due to the amount of vaccination. After that, they gradually started to decrease over time. So in 1946, the government of the country concluded that the vaccine was successful, discussed the evidence shown in figure 2.24, and against this conclusion. Basically, like I said, before the vaccination, the number of cases remained high and they gradually kept increasing. After the vaccination, there was a huge pike drop, and then they began to gradually decrease the number of cases over the years. All living organisms excrete waste products. Figure 3.1 is a photomicrograph of Naegleria fowleri, a single-celled protist that lives in water environments. Okay, so this is the structure or photomicrograph of this organism. State a feature of this visible in figure 3.1 that distinguishes it from prokaryotes. So whenever you hear the word prokaryotes and you need to distinguish a feature, prokaryotes, what is the opposite of prokaryotes? Eukaryotes. What's the major difference? Nucleus. Prokaryotes have no nucleus, eukaryotes have nucleus. The nucleus is present. Say the name and function of structure A. Structure A is a cell membrane. So the function of cell membrane is the controlling the movement of substances and molecules inside and out of the cell via diffusion, osmosis, active transport, and phagocytosis. Suggest how this organism excretes carbon dioxide. Carbon dioxide is excreted because the molecules are larger, so they are excreted via diffusion through the cell membrane. Urea is a toxin that is excreted by the kidney in humans. Describe how and where in the body urea is formed. So urea is formed by deamination of amino acids. Amino acids have the major nitrogen group unit, and deamination is a removal of this unit which results in urea. It is formed in the liver. Figure 3.2 shows parts of the human excretory system and associated blood vessels. The arrow indicates the direction of blood flow. Okay. Draw a label line and the letter X on the figure 3.2 to show the location of the cortex in one of the kidneys. Table 3.1 contains statements about the label structures in figure 3.2. Complete the table by stating the name of the structure, identifying the letter that labels the structure. So the cortex is somewhere here. Now, the completing the table, the description, name of structure, and letter. So the organ that stores the urine, without even looking at the diagram, this organ that stores urine is the bladder. The organ that carries urine out to the kidney is the ureter. The blood vessel that with the lowest concentration of urea is the renal vein. Lowest concentration of carbon dioxide, renal artery. Tube that contains the urine out of the body carries urethra. So after you've answered these questions without even looking at the diagram, now you can refer back to the diagram. G is the bladder storing urine. H is the urethra carrying urine outside. F is the ureter. Okay. E is the renal artery, and D is the renal vein. Our last question. Doctors wanted to investigate the effect of exercise on the excretion of salts. They collected urine from people before and after running a long distance on a hot day. The results of their investigation are shown in Table 3.2. So the average volume of urine per centimeter cube before running was 1,156 and after running was 569. The average concentration of sodium in urine per mole per decimeter cube was 85.6 before, after running 78.2. So suggest why there's a difference in the volume of urine produced before and after running. Well, obviously because after running there is excess amount of salts that are released in the form of sweat to maintain the homeostasis level. This is why after running there is a less amount of concentration and volume in the urine. Calculate the percentage decrease in average sodium concentration after running compared with before running. So average sodium concentration is what we're looking for. Before running was 85.6, after running 78.2. The percentage decrease formula is a negative percentage decrease is equal to minimum minus maximum divided by maximum times 100. Substitute the values in. Minimum is 78.2 minus 85.6 divided by the maximum 85.6 times 100 is equal to 8.6. So which is approximately negative 9%.